Our next speaker is Neil Gural from Harvard and MIT. She couldn't be here today, but we have her work in Sangeeta Bhatia's group on a microengineered liver platform will instead be presented by Dr. Sandra March, and she's the head of the malaria team in Sangeeta Bhatia's lab, and she's worked with her on this project. So thanks for coming. Here she is.
Okay, so good afternoon. Thank you for the invitation to share with you our laboratory recent efforts. So, as you know, the liver stage is an attractive startshed for the P-Vivax eradication. However, this stage is still understudied. The reasons are that getting access to these parasites is challenging. Most of us, we need to travel to endemic areas to perform our experiment. In our case, to Thailand, where we work in close collaboration with Dr. Yetsuman. Another reason is that P-Vivax is human-specific, so human-living platforms are needed. So our laboratory have developed a human liver platform that can recapitulate the full life cycles of Plasmodium falciparum and Plasmodium vivax. Here I'm showing a schematic of our platform and our infection protocol. This in vitro system is based on aquaculture of primary human hepatocytes that are seated on islands and surrounded by a supported stromal cells. In that way, we stabilize the functional hepatic phenotypes for up to 3 to 4 weeks. Our primary readout is imaging. Okay, so using this in vitro system, we have characterized the development of these P-Vivax parasites. Here we can see ymnozoites and developing schizoin at 3 different time points. At day 8 post-infection, we can observe clearly 2 populations, big and small parasites. And at day 21 post-infection, mainly 1 population of small parasites. So, at day 12, we could observe in real time the release of merocytes. Here in this image, we can see 3 schizoins that contain inside merocytes with the white arrow. And then we can see 1 merocyte that is releasing, 1 schizoin that is releasing merocytes. And we are going to show a video showing this event. So you can see here, these are the schizoin, and this is the one schizoin that Neil could capture that it was at the time releasing merocytes to the culture. I mentioned that our cultures are in the 96 well format. And finally, we demonstrate that these merocytes were able to infect overweight reticulocytes. The infection rate was approximately like 3% of the reticulocytes that we added that were infected. So, although in our culture we observed small forms, we wanted to make sure that in fact they were ymnozoites. So, we performed a characterization of these small forms in our cultures based on a previous hallmark described. The small forms observed in our cultures were mononucleated and persisted after 21 days post-infection. They didn't express late-stage markers, like MSP1, and some of them spontaneously reactivated. You can see here a big schizoin at day 21 expressing MSP1. And finally, we demonstrate that these small forms were resistant to drugs other than primaquine. So we have here a culture with primaquine where we could achieve an IC50. However, when we use our cultures, we add atovaquone. We couldn't achieve any IC50. We saw a decrease in the number of sporocytes, even a highest dose, but we couldn't achieve an IC50. However, we perform a segmentation of these parasites in big and small parasites. we could observe how otobacone killed all the schizones, but the ipnozoines, the small forms, were resistant. Next, we use the platform to assess the activity of a set of drugs in clinical development in a prophylactic and radical cure mode. So as you can appreciate, the activity of most of the drugs tested in the radical cure mode was lost against the ipnozoites. So our laboratory, with the goal of a better understanding of the biology of these ipnozoites and ultimately to discover new drugs, we sought to uncover the transcriptome of these ipnozoites. We performed an RNA-seq on ipnozoites-enriched MPCC cultures on day 9 post-infection. Ipnozoites enrichment was achieved by treatment with a PI4 inhibitor in a radical cure mode and processed in parallel with an untreated culture, referred here as a mixed population. So total RNA from infected MPCC was prepared, and we performed an enrichment for P-virus transcripts by magnetic pulling down using custom-made beads. Selection by hybrid capture was followed by Illumina high-seq. These are the quality controls. We got an average of 29 million reads per sample. An alignment to P-Vivax P01 transcriptome revealed robust enrichment with approximately 19 to 48 of the reads mapping uniquely to P-Vivax in mixed and ipnozoite cultures, respectively. So it was a good enrichment. So here we can see the transcriptome that are different from the ipnozoites and the mixed population where we have the untreated cultures. So looking at gene expression, we identified 880 genes that were significantly suppressed and 42 genes that show increased expression in the ipnozoite-enriched sample as compared to the mixed samples. So here we have 880, and then we have 440 here in this group. While 40% of the identified transcripts encode a protein of unknown function, the gene list contains RNA and DNA binding proteins, nuclease, protease, and transferase, suggesting a preserved metabolic and catalytic activity, which could be linked with the increase in size that we observe in our schizome from day 8 to day 21. Next, so this is the, I presented, so the previous, this is the transcriptome data, bulk transcriptome data we just recently obtained. We are still in the process to analyzing, validating some of the hits, and next we want just to perform a time course because this is only from the 2 time points. We will perform a kinetics. But what we want just to do next, and now is in parallel with all the analysis that we are currently doing with the bulk transcriptome, is that in our laboratory now in collaboration with Alex Shalek at MIT and Dr. Yetsum at McGill University, we are also pursuing single-cell transcriptome of the parasites using sequel. By doing that, there are like 2 main advantages compared to the bulk transcriptome, is that we could attempt to capture the potential heterogeneity of these ipnozoites, and also important, the response of the hepatocytes to the distinct liver stage. So it's going to be like a dual transcriptome of the parasites and the hepatocytes. This is a very preliminary data. It's a proof of concept, so we still have a lot of work to do that, to be done still. But we have already performed the first pilot experiment where we could generate 84 transcriptomes of individual P-virus parasites. Using a tcDNA analysis, we obtained 3 clusters. And based on our bulk transcriptome data, cluster 1 and cluster 2, the ones in green and the ones in blue correspond to the schizone and an ipnozoite stain. And then we think that we found another cluster that is cluster 0, and that we think that they are the reactivating ipnozoites. as these clusters share genes found in both hymnozoites and schizones. So as a summary, the MPC platform supports the formation and reactivation of hymnozoites. P-virus schizones see the MPCC mature, release merosomes, and infect reticulocytes. MPCC allows prophylactic and radical cure testing of anti-hymnozoite drugs. And using this experimental platform, we have provided the first transcriptome of P-virus hymnozoites. I would like just to thank Sangeeta Bhatia and especially the malaria team, but Neil is the one that have been done most of the work presented here. And with Liliana also, it's played an important role with all the bulk transcriptome data we presented. We also would like just to thank our collaborator, Dr. Jet Soman, and her team at the Mahidol University, and our collaborators in the core that we have at MIT that help us with transcriptome, and all the other collaborators that have been participating. Thank you very much.
Any questions for Dr. March? Don't be shy. Yeah, so.
Really interesting presentation. Thank you so much. The effect of atovaquone was interesting. Did you know that it would not kill hypnozoites when you selected it, or did you discover that?
No, we knew it. So it was a way to prove that in our cultures we have hymnozoites. It was already described that atovaquone was not going to kill hymnozoites.
Where is that described? How did you know that?
Well, it's always what we have heard in the community. Not atovaquone, it's like what's not going to kill, and it's what's happening in our hands.
Okay.
Clinically been shown that hypnozoites not kill like.
Yeah, there's some clinical evidence, but it's not very good. And there's some clinical evidence that says actually it does kill hypnozoites, and some people actually believe that. Clinicians, some do, but yeah, it's very lovely to see that you've convincingly showed that it absolutely does not kill hypnozoites.
Yeah. So in fact, yeah, correct. It was not something that we were surprised to see it.
Yeah, so I was just wondering, when you're following the cells in culture and you show the day 8 and the day 21, and clearly at day 21 you have the small forms, but is there a time along that when you have a clue as to which ones are reactivating or turning, like is there a point of divergence? When's the earliest that you guys can tell that you feel like some are schizons versus hypnozoites, or is it really just based on the atovaquone treatment?
No, well, at day 8, so it is clear we have two populations that you can see, right, small and big forms. So we are like sure that these small forms are the hymnozoites. Yeah. And the reactivation takes some time, so we don't know really at later time points. But at day 8, so it is we have the confidence.
Right. And you mentioned that you see that they spontaneously reactivate, and there's nothing that you all do or see different. I wondered about the divergence between different strains and different experiments.
Correct. So all these experiments we have been doing with in Thailand, so we cannot compare with other research, and we don't know. So it is spontaneous. So we would like to know what we can do to reactivate them, because it could be, we can think that reactivation could be a different strategy just to kill these hymnozoites. So right now it's quite difficult to kill these hymnozoites. We only have one drug, primaquine, but what about if we try just to wake up them and then use other drugs that we have? We have many others to kill the schizons. So, but right now we don't know how we can reactivate these hymnozoites.
All right, thank you. Thank you so much.
Thanks.